Okay, so this is a slide, if you remember. 1 minute. Oh, sorry. So sorry. Yeah. Yeah. So if you remember this particular slide, so you need, the normal or the and the disease samples. Nevertheless, if you are using on a bacterial samples, you might need a wild type and a treated sample as well. Okay, and we also discussed, why do we need what you call um, um, why do we need to do a RNA-seq? Why can't, we perform probably using a microarray or a simplest, qPCR? As I told you, qPCR has a limitation. You cannot go for, n number of samples or with n number of, organisms, per samples. Either you have to go for multiplexing, even that has got a limitation. These days, you may go for 1584 weld plate, which is a very cumbersome process because you need to raise, primer probes, for each of those particular, genes of interest. Whereas microarray, although it is better, the kind of, probes that would probably be used for microarray is again limited. If at all, if you were asked to identify certain unknown gene or known unknown gene, where you know what that particular gene is, but you don't know what the function is. But those particular things, you may not really have a probe, that is really fitting in for microarray. Okay, be it for Agilent or Epigenetics chipset. Okay, and that is again a limitation. Whereas in a whole transcriptome shotgun sequencing or RNA-seq, you don't have that particular limitation. So you can measure the transcript abundance at your goodwill for n number of samples. So for each of those particular samples, you use something called, cufflinks command. Okay, where we have benchmarked our pipeline, which I'm not going to give you because it's an abridged version. If at all, if you have, multiple number of, replicates, you can probably use, cuff merge. Okay, so that is where, you could probably use, cufflinks, cuff merge, and cuff diff command. Okay, so here, what I'm just trying to tell you is you have to use cufflinks, and that SAM or BAM file you can use. So you run normal FastQC, then you go for HiSat2. HiSat2 is used, to check the, splice junctions. Okay, those wouldn't probably be missed. Those particular, regions wouldn't be missed when you run HiSat as opposed to Bauti2. Okay, and then you come up with something, what we call it as, this cufflinks. I mean, you can even SAM tool sort, where you'll sort the BAM file, and that BAM or SAM file, you can use it, using this particular command. And when you run this, you will get, a list of, files. What are those list of files, which I'll probably again show you using cufflinks. So these are all those cufflinks, things you'll get. You'll get some assembled transcripts, some script transcripts, okay, in the form of a GTF file, which is a gene transcript file. Okay, this is for your individual samples, and this is what, we have used, for our collection data set. Okay, and then you run the final step, which is cuff diff. Okay. So as I told you, for cuff diff, this is the syntax. Okay, so you run transcripts.gtf. Is my screen visible? I hope it is visible, right? Yeah. So you use transcripts.gtf, which is the transcripts emanating from your control sample first. And then the sample 1 is again your control. Okay, and sample 2 would be your treated. So if you give transcript.gtf for your control, which is your control transcripts, and then accord your SAM or BAM file for your control, okay, and then, equate it and find the differential express genes to that of a treated SAM or a treated BAM, then you'll get a list of, differential express genes. Okay, so this is how, the command needs to be given. Okay, and you cannot reverse it, which means you cannot start with, taking the inverse proportion. Maybe you cannot start with the treated GTF, treated SAM or treated BAM, and control. You cannot do that way. Okay, that's probably an unorthodox way of doing the RNA-seq approach. Okay. Whereas there are other approaches, like as I told you in the slide, you have got DSeq, EdgeR, and BioZar. Okay, the only difference between cufflinks, cuffdiff, as opposed to EdgeR, DSeq, and BioZar is EdgeR and DSeq and BioZar rely largely on the replicate ratio. Okay, that is a very important aspect, which means they work on dispersion estimation between replicates. So which means you'll have, say, sample1.sam, sample1a.sam, sample1b.sam. Those are all those particular individualized samples across those particular replicates. So if there are no replicates, absolutely, maybe DSeq or EdgeR, that Bayesian way of dispersion approach is nullified. They may not really run. That estimation is not quantified properly. Okay, and that is the reason why you probably use cufflinks, cuff-diff pipeline. It's a very simple, from a biologist perspective. If you want to merge replicates, you can use it. If you don't want to merge replicates, it's fine. And most importantly, you can use rigorous statistics, and you can also use normalization, as we were discussing, the FPKM, the fragments per kilobase of transcripts per million map reads, RPKM and TPM. FPKM and TPM are phenomenal. TPM is largely for those paid and reads, and where you can quantify the transcripts that are mapped across. Okay, whereas reads per kilobases per million map reads is largely a read. As I was telling you the other day, if you have this, okay, this is a read, whereas this is a DNA fragment. Okay, something like this. Okay, so that way you can probably do this. So what I was doing the other day was yesterday, not the other day, yesterday. I was running this cuff-diff. For some reasons, it was giving some error because it's a collection data set. It's because the collection data set was on 128. So when you look into 128, our sample, okay, where is that sample? Yeah, this one was the sample high sat to what? Sorry, 126, not 128. Yeah. So this is our control and this is our treated. Okay, SRX 19447548. This is in a collection. This should be split up. Okay, and then only you can run your cuff-diff. So you run cuff-diff like this. Okay, and then okay, and then here you take this transcripts. Okay, so usually, as I told you, there are two types of transcript. One is assembled and skipped transcripts that you get. It's still reading. 1 minute. Yeah. So here you take this. Okay. Assembled transcripts you can take. This is number 131. Okay. And here you give SAM or BAM, as I told you. If you remember this particular command. Okay. SAM or BAM for your control now. Okay. So now you do this SAM or BAM, and then you define it as a control. Okay. And take this data set collection and chose that. In fact, it should be individual, but it's not there. Individualistically, this ID is not there. Okay. If it were there, no. Otherwise, it has to select. It has to read this. Clear, all of you? Yeah. Yes, please. Yeah.
Sir, if we are having more than two, means one control and 3 or 5 samples, then
yeah. instead. But if they are from the replicates, you can merge them. You can merge using cuff merge. Okay, but if they're individual proportionate, then you can run cuff quant. Cuff quant also comes in a similar way. So it is like this. You run like this cuff quant. Okay, so you see it's the same thing. You can quantify those replicate SAMs across each condition. So you can do this. Okay.
Thank you, sir.
Yeah. Oh, sorry, I didn't share the screen. I'm sorry. I thought I'll quickly share 1 minute. Yeah. So this is what cuff quant. This is what I was showing you. So it's you can use cuff quant like this in a command line, but you can find those replicate SAMs and you can give comma replicate one, replicate two, and so on and so forth. Okay. Yeah, so we'll reserve some questions later. So I've already introduced Rohit, Reja, and it's 6:32 now. So we'll have an exciting session now. It's absolutely a novel topic that Rohit has been working on. He has been working in these frontiers areas of NGS and systems genomics over the last 14-15 years, and his expertise would largely be helpful. So please have a very good session and interaction today. Over to you, Rohit. Thank you so much.
Thank you, Prash. Let me share my screen. Um, can you all see my screen? Yes?
Yeah, yes.
Okay. Before I begin, I'm going to put the link to the Google Drive in the chat. This link has all the data set that we'll be using for the workshop and also this presentation. And yeah, please feel free to stop me at any time during the presentation to ask questions. This is an interactive workshop. So first of all, I want to give a shout out to Prash. Thank you so much for giving me an opportunity to share some of my experiences with the analysis of this kind of data set. And since this is the first time for me to talk to all of you, I just thought I'll just give a brief introduction about who I am. So I grew up in a small city in Madhya Pradesh, and for my undergraduate, I went to VIT and with bioinformatics. And this was really initially when I actually came in contact with Prash when he was in Denmark, and that's when I feel like I really started a bioinformatics project, and we collaborated on a hypothetical proteins, hypothetical genes project. That was kind of my first foray into any bioinformatics analysis. So I'm really thankful to Prash for that. After I finished my undergraduate, I actually went to a Korean Bioinformation Center in Korea as a research assistant. And before you ask, it was not North Korea. So after a brief stop at EMBL EBI, where I did a short internship learning text mining and mining the PubMed datasets, I ended up in Penn State, where I did my PhD. This is where I worked with Dr. Frank Pugh and focused on understanding the gene regulation, utilizing a lot of these ChIP-seq, ATAC-seq technologies, these genomic assays to really understand how a gene is really transcribed and how the transcription factors arrange themselves on the promoters or enhancers and how they regulate the expression of the gene. And after my PhD, I moved to San Francisco, and I have been here for the past 7 years. And currently, I am in a company called Genentech, which is a pharmaceutical company where I am a principal scientist and group leader in oncology bioinformatics area. So the learning outcomes from this workshop is I hope that by the end of this workshop, you will be able to analyze the ChIP-seq and ATAC-seq data sets. You would learn different downstream tools that are actually commonly available for the analysis of such data sets, and you will get familiar with them. And lastly, you will also learn about different experimental designs that should be taken into consideration before you actually start on your ChIP-seq, ATAC-seq experiment, because that's really important that you have proper controls, you have proper replication of your experiment. So this is the outline. First, I'll just go a little bit into what ChipSeq technology is. What are the different assay considerations that you need to be thinking about? And then I'll give you a brief overview of the analysis pipeline. And then once we go over the pipeline, I'll give you an example of how we can apply ChipSeq to interrogate biological processes, how we can apply ChipSeq to actually understand a biological question. And then a little overview about what are the different advancements that have been made in ChipSeq so far and how that's actually improving the resolution. After that, I'll jump into AttackSeq and a little bit comparison with ChipSeq, and then an AttackSeq example, followed by what are the different advancements we have in AttackSeq technology. And then after all of this, there's going to be a workshop where I'm going to show you the different downstream tools that are mostly online tools that you can use for downstream analysis. And I have a lot of little data sets in the Google Drive folder that I've shared. Feel free to download all of those files onto your computer. Okay, so let's begin. So as you all know, eukaryotic transcription, it's a very complex process. And let me get my pen here. No. Sorry, 1 second. Doesn't give me the option. Anyways, this is fine. So as you all know, eukaryotic process is a complex process, right? So first of all, the DNA is actually wrapped around something called as nucleosomes, which is essentially is formed by 4 histone subunits. This sort of wrapping up of DNA around the nucleosome allows it to be packed in such compact structure and be folded inside and be form this compact structure, and that's how it resides inside of a nucleus. The nucleosome that is downstream of the transcription start site, or TSS, is called as +1 nucleosome, and the one that is upstream is called as -1 nucleosome. Now, if you just look at the region between the +1 and the -1 nucleosome, you can see that you would find there a gene, which is shown here with this intron exon boundaries. And then upstream of it is a promoter or the promoter proximal element. For a gene to actually be able to transcribe, this region needs to be opened up. So a region might reside in something called a heterochromatin, which is basically when the DNA is packaged like this, and there is no way a transcription factor can go in here and start the transcription. So for the first process is actually the DNA element to become available, so that nucleosomes have to slide down, slide up, and make this region accessible. That's when any transcription factor can really come in and start the transcription process. So if you think this is complex for a gene to transcribe, there is another elements there called as enhancers. And these enhancers, they interact with the promoter regions through some proteins, and they form these DNA loops. So not only the gene expression is controlled at the promoter level, but it is also now controlled by multiple enhancers. So you can imagine that's just to really tear it apart, how a gene is actually getting transcribed, not just by a promoter, but also by different combinations of enhancer. So to really interrogate this system, we can basically look at one gene at a time. And this is the +1 nucleosome, the -1 nucleosome, and we can use PCR, qPCR approaches and whatnot and study, okay, maybe what's going on here, what transcription factors are here, just understand it. However, just looking at just one gene at a time, we cannot extrapolate to it really what's going on at other set of genes, because each gene might have its own different configurations, its own unique mechanism of regulation. And we can't really figure out just by studying one gene at a time. And that's where genomics comes in, because with genomics, as Presh was mentioning before, you can really examine hundreds of genes at the same time. You won't have to rely on just one gene at a time, and you can apply these next generation sequencing approaches to really interrogate not only one system, but multiple systems at the same time. So that's really where the power of genomics comes in. Um, so there's an organization called ENCODE, which stands for Encyclopedia of DNA Elements. And ENCODE has been in place for the past 10 to 15 years. And ENCODE basically came into place right after the Human Genome Project, where now people thought, okay, we have figured out the genome, but they realized, well, the genome is far more complicated. And now we need to understand how different, functional aspects of genome are organized. And so ENCODE really came up with a bunch of different assays. Some of them we are already familiar with. For instance, RNA-seq, which is more of a functional genomics kind of an assay where you're trying to understand, how much of a transcription a transcript a gene is producing, how less of a transcript a gene is producing. So that's one of those assays. There are approaches like ClipSeq or RipSeq, which understands how different RNA binding proteins bind to RNA. There are approaches like 5C or ChiaPet. You probably might have heard of these as well. These are to interrogate the DNA loops that I was talking about, these chromatin structures like the enhancer promoter binding. And obviously, assays like ChIP-seq, which I'm going to be talking today here, but also technologies like ATAC-seq, DNA, and FAIR-seq, which is that a question? Sorry, I
No,
can't.
sir.
Mhm.
Participants are asking just to be a bit louder, sir. That's it.
A bit louder.
Yes, sir. Please.
Okay.
Thank you.
Okay, let me try. All right. Let me know if this is okay. So there are different technologies that ENCODE has suggested, has come up with, and one of them is ChIP-seq, which we'll be talking about today here, and then the others are ATAC-seq, DNA-seq, and FAIR-seq, which are to interrogate the open chromatin in the region to understand which regions of the genome are open, as I was telling you before. Um, all right. So let's jump into what ChIP-seq is. So ChIP-seq is a really powerful tool to investigate genome-wide where a particular protein or a transcription factor, TF in this case, is binding in the genome. So to do that, you start with cross-linking step where you cross-link your proteins that are bound to DNA onto the DNA. So now this particular protein is cross-linked onto the DNA, followed by sonicating the DNA. So now you have different DNA fragments where you might have your transcription factor of interest bound and you might have your nucleosome bound, followed by a enrichment step, which is used where an antibody of interest is used. So for instance, if you're interested in a transcription factor, you would use an antibody against that transcription factor to enrich for those DNA bound fragments. So for those protein bound DNA fragments. So this way, using an antibody against, let's say, CTCF transcription factors, we're enriching for those DNA fragments that are bound by only by CTCF, not by nucleosome or anything else. And once you have these DNA fragments, you then reverse cross-link, remove this protein here, and then take those DNA fragments, attach Illumina adapters, and just like in your RNA-seq experiment, you would sequence these, and you would get FASTA-Q files. Then you go on to the pipeline process. You build this sort of an enrichment profile and essentially call peaks, which basically are the regions where you have a lot of enrichment of these DNA fragments compared to a control setting. In short, this is basically what a ChIP-seq protocol does. And you can see that there are so many steps that are involved in this process. One of the considerations that that we should really take for ChIP-seq is always having a negative control, which could either be an input, which would basically be just a sonicated DNA, or an IgG control where the antibody is against IgG. So it's basically against something that is not part of your transcription. It's not a transcription factor. So basically, that way you have a control to always compare against. So that's always having a negative control. And second, having replicates. So you can always you should always go for at least 2 to 3 replicates per condition. So for instance, you're doing a transcription factor, CTCF, for instance, and this transcription factors you're going to do in a untreated and a treated condition, and you want to understand if there are changes in the binding site in the treatment compared to the untreated. If you only use one replicate for each condition, you would not be able to do any of the differential peak analysis downstream. So it's good to have at least two replicates per condition so that you can at least assess how well your system has worked, how well the ChIP-seq protocol has worked, and if you can do some of this further downstream analyses. All right, is my voice still okay for everybody to understand?
Yeah, perfect, Rohit. Yeah.
Okay, so this is the ChIP-seq pipeline. And here I'm saying you start with mapped read because the process of taking the FASTQ files and using BWA or Bowtie to kind of map your reads are kind of pretty much the same. That process does not change. When you have your mapped reads, that's where you actually start the ChIP-seq pipeline. So you go through the process called peak calling, which is basically looking for enrichment of your transcription factor bound DNA across the whole genome, and comparing it against a control sample. And there are different tools like Max2, GeneTrack, and GPS, and I'll talk about these tools in the following slides. But just to give you, just the steps, what are the steps that are involved? Then we move on to the step called as removing blacklisted region, which you can probably figure out from the name that these are blacklisted regions, and so they should be removed from and what those are, I'll cover those as well. Then we go on to, if you have replicates, we look for the concordance between the replicates to see that both replicates are actually giving you almost similar output. And then we go on to the downstream analysis, which we'll cover this in the workshop later on, which is to perform motif analysis using these tools called MEME and FEMO, and using some sort of an ontology analysis using a tool called GRADE, and then visualizing your data sets, either in genome browser or using certain other tools as well. So this is basically a very broad pipeline. Using this pipeline, you can really figure out exactly where your transcription factor is binding, what motif it recognizes, and also what kind of genes it regulates functionally. Okay, so first I'll move on to the peak calling step. So essentially, the peak calling step, as I mentioned, that when you have a transcription factor bound DNA fragments, you can imagine you're sonicating your DNA. So you'll get these random fragments that are around the transcription factor, right? So you'll get some of these fragments. You align these sequencing tags, you align the forward reads, you align the reverse reads, and you get some sort of this enrichment around the transcription factor binding site. So this is where the transcription factor was bound, and obviously this region will not be, will be saved from sonication. So you'll get these fragments that are, that are around the transcription factor site. So peak finding step is basically intuitively is trying to find that midpoint through different approaches and moving those reads to this particular midpoint. So you can say, okay, this is where the transcription factor was bound to the DNA. So Max2 is a very popular tool. This is most frequently used a tool for ChIP-seq peak calling. This is also a part of Galaxy tools that Prash was showing you so you can take your BAM file and run Max2 through Galaxy. The way Max2 works is basically it tries to estimate a shift distance. So basically tries to see how much tags need to be shifted based upon the distribution of these reads. Once it's kind of estimates that shift distance, it shifts those reads onto this particular midpoint. And then it kind of builds the stack density. And then based upon the stack density, it estimates the local maxima in that particular peak. And then sees compared to the input control, how much of an enrichment here that there is. And then it basically makes a call whether this should be called as a peak or not. Essentially, a peak would have a high enrichment at certain positions compared to the input control. The next tool is GeneTrack, which is also commonly used tools, and also kind of works very similarly to what Max2 does, but instead of looking at, it also kind of builds this density of the, on the positive strands and the negative strands, and then it pairs each peak that each local peak that is there in the positive strand with the negative strand, and kind of tries to estimate what is the most common shift distance that we can estimate, and then based upon that, it kind of estimates, okay, this is a shift distance that we should put all the tags to, because that's most likely where the transcription factor is binding. And lastly, GPS. So I haven't used much of GPS, I'll be honest, because I haven't had a lot of experience with it, but it seems a little bit more statistics oriented, where you start with just randomly positioning your binding site, and then you build a model around the binding site. Then you, based upon the actual tag distribution, you really estimate how well that model fits into the actual distribution, and then you iteratively move this binding point to a certain location until the model really fits well with what your actual data is. And that's how you kind of estimate where the binding site would be. Um, Irrespective of the tool choices, what you get at the end of the running these is basically a list of different peaks, basically, which would be basically chromosome, start position, end position, and the enrichment value. It's going to be in a bed file, which we're going to talk about later in the presentation. But it basically tells you these are all the positions in the genome where we think there is a transcription factor that is bound. Okay, so now you have your peak called. The next step is actually to remove the blacklisted region. Now, that's really very important concept in ChIP-seq and ATAC-seq technologies. So what are blacklisted regions? So as you can imagine from the name, these are blacklisted regions. These are blacklisted for a reason. So blacklisted regions are the regions where, irrespective of what transcription factor you're using, whether it's even control, those regions are just very non-specific, and they comes out as very, very heavily enriched. So for instance, this is an example from this particular paper, where if you look at the blacklist region compared to a normal region and look at the read distribution, they have a lot of reads that actually come out from those regions compared to a normal blacklist region. Compared to a normal region. So you can imagine that if you're doing a peak calling, if you're doing any kind of normalization or whatnot, this region would actually create a lot of problems. And another example is a IGV screenshot here, where we're looking at these BAM files, or these big files, and we're looking at a blacklisted region. And you can see it doesn't matter if you're looking at the polymerase 2 ChIP-seq data or MIC ChIP-seq data. This region looks exactly the same, has exactly similar sort of pattern of binding, kind of indicating this is a non-specific region. It is not a real peak and should be removed from the analysis before we actually move on to other downstream analysis, because otherwise it might interfere with actual data. So these regions are really removed from the downstream part of the analysis. Okay, so now you have removed the peaks, you have removed the blacklisted region. And as I mentioned before, ideally you would have at least two, if not two, three replicates. But generally, most of the people stick with two replicates because ChIP-seq is actually quite cost intensive to do. And so most of the people stick with just two replicates, which is fine. And now the next step is actually to assess how well both of your replicates are. And ENCODE has actually come up with this thing called as irreproducible discovery rate method, or IDR method. And they have a whole framework around it to actually figure out whether your two replicates are actually good or not. And so just to give you an intuition about what this framework is, and there's a whole bunch of metrics that you calculate to really come up with whether it's good or not. But intuitively, this is basically what the method says. So you can imagine that if you have two replicates, and for both of the replicates, if you have the peaks that have a really that have a lot of signal in both of those peaks, right? If they have a good signal in the peaks, that most likely represents that those are bona fide peaks. Those are actually real peaks, compared to the peaks in the data sets where the signal is not that strong compared to the background. So if you now plot one replicate on the x-axis and the other replicate on the y-axis, and you just plot the signal value, you can imagine that these black dots are the ones which are high intensity or high signal region. You can see that this is a very sharp distribution between the two replicates. That means if there were really bona fide peaks in the one replicate, they would be most likely bona fide in the other, and they would form this kind of this sharp pattern in black here. However, the peaks that are not as strong would would be a little dispersed like this. They would be maybe stronger in one but weak in the other, and they would be here and there. And you can probably say maybe those are not real peaks. Another way of looking at it is if you actually rank your peak, from the most strongest to the least strongest, you can basically see the same pattern where you have a bunch of peaks that are more reproducible. Those are the ones, the bona fide peaks, versus the ones that are more dispersed distribution. This is actually an example of a really good data set where you have good concordance between the replicates. However, if you look at a bad example, you can see that these black dots actually do not, there's very less of those, where these are the peaks which are really strong in both replicates, and you can see that these are pretty much non-existent. And majority of the peaks that are really there are these this in the dispersed region, which are the peaks that are only present in one data set versus the other. So using this to kind of as an example, ENCODE came up with this IDR framework, and to basically assess how good a replicate is. And I'll probably walk you through this, but there is a whole tool out there that actually does this work for you, so you don't really have to individually do this each step at a time. But I wanted to sort of give you this framework because that's a framework that is actually used by ENCODE to assess the replication between the two replicates. And basically, the steps are that once you have these two replicates, you actually assess the IDR between the two replicates, basically by kind of plotting them this way and basically assessing this point of deflection between the two replicates. So that's how you kind of calculate this one metric. Then you take your replicate, both your replicates, you pool them together, and then you split them again randomly into two pseudo replicates. The reasoning behind it is that if there are really good peaks between the two replicates that are reproducible, mixing them together and then randomly sampling them again into the pseudo replicates wouldn't really change the peak strengths. They would probably appear in both the pseudo replicates as well. And so then you make these pseudo replicates, you do this IDR calculation, and you get another metric as well. And then thirdly, an optional step is basically you can take a replicate itself and split it randomly into two replicates, and then calculate this IDR metric as well. Basically the same concept. The high intensity peaks will be basically the same, and then you'll get another metric. And then using the combination of metrics, then you can basically assess whether these are high reproducible replicates or low reproducible replicates or not. But essentially, that's basically the core idea behind it, is you're looking at those high intensity peaks and making sure that they're actually present in both of your replicates. Okay. All right, so that was basically all the steps, and essentially, basically, the first three steps are trying to make sure that you're looking at really good peaks, you're looking at the peaks that are reproducible, and you're looking at the peaks that actually belong to that particular transcription factor. The next analyses are actually quite fun because now you actually really get to discover what those peaks are, what genes do they regulate, and if you can really visualize those peaks, and see where exactly they're binding on the genome. And we'll be doing these in the workshop as well, so you'll actually see how that data looks like. So the first tool is for figuring out what DNA element does a the transcription factor binds to. And the DNA element is usually a sequence of basically just a nucleotide sequence, which is defined as a motif. And each transcription factor has a very unique specificity to a particular sequence of DNA elements. And the way you do it is using this tool called as a Beam Suite. It's a web-based tool, but also you can run it command line as well. And the two tools that are most commonly used in this whole suite are the meme and the meme chip, which is basically used to, you give it your set of peaks, the faster, to give you the faster file, which is basically the sequence within your enriched peaks. And it basically looks for over representation of a particular DNA sequence within those in peak regions and gives you maybe what are the different peaks that are actually, what are the different motifs that are enriched inside those peaks. You can then take those peaks, those motifs, and submit it to TomTom, which is a database of known motifs. And so then TomTom would actually tell you, okay, this motif seems to be a CTCF motif, or this motif that you found could be transcription factor XYZ motif, which is really good to really recognize what that motif might be, are unaware of what that motif is. And FIMO is then takes that motif and does a genome-wide search and tells you exactly where in the genome that motif is enriched. So now you can actually really see within your peak where exactly that motif is present. So this is a really good collection of tools, and I use it very frequently and very useful to quickly look at your ChIP-seq data set. The next is the ontology analysis tool. So essentially, what you want to know is if within your peaks, if you have a set of peaks, which genes they are regulating. And sometimes these peaks could be binding at promoter. So it associates those peaks to the nearest gene promoter. Sometimes your peaks could be binding to the enhancer, and it basically assigns the nearest gene to that enhancer as well, to then get a list of genes, which it then does an ontology analysis to figure out, what is the functional significance of this particular transcription factor. And lastly, so this is an example of the tool called GRADE, which gives you the first, it tells you basically, if you give it a set of peaks, how far from the transcription start site or close to the transcription start site those peaks are binding. So this is an analysis I did for P300 transcription factor, and it tells me that P300 transcription factor binding sites are actually 50 to 500 kb away from the TSS, which to me tells me that these are mostly enhancer peaks. There are a lot of enhancer binding peaks for P300. If they were promoter peaks, they would be within 0 to 500 kb of the transcription start site. And these ChIP-seq peaks are actually from mouse embryonic forebrain, midbrain, and limb tissue. And so looking at the ontology analysis that the tool gave me, it says, oh, this transcription factor seems to be involved in tube development or limb development or morphogenesis. So that's sort of suggests, okay, this transcription factor plays a role in these embryonic development of these different limbs, and that's kind of a really a novel insight that you can actually take in design a further hypothesis. And the last step is actually visualization, and there's two tools I'm going to mention here quickly. One is a tool called as Deep Tools, and this is essentially a collection of different functions. I probably won't be giving a demo here, but I did want to bring this up to all of you because this has a really good collection of different kinds of plots that you can make. And one of the plots that I'll also talk about in the slides in the slides further, but you would probably kind of see these kind of plots in a lot of ChIP-seq papers, and I thought I'd just maybe briefly touch upon what this really means. And so this is a plot where it's sort of a heat map where each row here represents a different enhancer region, and this is centered across the peak of the enhancer. And so you're going around 2 kb upstream and downstream from the peak center of this enhancer, and you're lining up all of these enhancers. And then you're coloring up each base pair based upon how many reads there are. And the more reads there are, the more blue the color will be. The less reads there are, the less, the more red it would be. And if you sort them basically, by the number of reads that are from top to bottom, you get this nice distribution, which tells you the top peaks here have a lot of enhancer. This is actually a DNA signal, so actually tells you that a lot of these enhancers that are at the top have a really good open chromatin. As you go down towards the end of these enhancers, that's where the chromatin is actually closed. And you can sum all of these regions up, the intensity in this region, and it gives you this nice composite plot, which then you can summarize and say, okay, this is where the peak strength of the DNA signal is. So really good set of tools. There are other tools that are part of this deep tools package. So I would highly recommend sort of exploring this package as well. And lastly, I'll also talk about this WashU epigenome browser. And it's basically a genome browser like IGV, which you might have used in the past for looking at your VCF files or your BAM files. And it is basically to look at your peak files now. It's basically kind of looking at those BAM files and then looking at how your peak. So this is how a peak would actually look like. And these are the different, let's say, different replicates. And you can say, okay, this is where this is where my peak is for the HOX9A gene. This is how it's probably regulating. This is probably binding downstream of my gene. This is binding upstream of my gene. So that gives you a lot more insight about a specific locus that you might be interested in for your analysis. So these were a bunch of different downstream tools that are very commonly used for ChIP-seq analysis. And now I'm going to jump into about how we can apply ChIP-seq to interrogate our question of interest. But before I go there, I just wanted to take a pause and just see if there are any questions. If I went too fast at some point, if you might like to repeat up any of the slides, I'd be happy to do that. So if there are any questions, please let me know before I move on to the next part.
So thank you so much. It was a very good lecture, actually.
Yeah. Do you have any questions? Anybody? If there are no other questions, maybe. Yeah, I can then move on.
Right now, we don't have questions left.
Okay.
Yeah, can I ask one question, Rohit?
Yeah, sure.
Yeah, wonderful talk, Rohit. So I was just wondering. So is it like into the open chromatin elements and their propensity? So can we assume that more the girth of that particular open chromatin element graph, greater is the chromatin content present in that particular sample? Is that how we look at this figure that you were showing earlier?
So they look at that's a great question, Prash. So look at the intensity. So basically the signal, the amount of reads that there is in. And for ChIP-seq, it's basically you're looking at the amount of reads in this particular and then that's basically your signal strength. And the more the read there is, the more you can imagine the stronger the binding of the transcription factor to that DNA region. And the basic concept behind is it's essentially a transcription factor that binds to a DNA. They're always, like binding and coming off from the DNA. That's how they kind of interact. The stronger the binding, you can imagine the more residence time of the transcription factor would be on the DNA. And in a population, you would probably, the more residence time, the more cross-linking that would happen, and the more you would actually see the reads coming out from that region. If the binding is not strong, you would have a lot of, like binding and removing, and that's when you see a lower strength. So that's basically the logic that goes into more reads, the more stronger the binding is.
Right, wonderful. And this browser, so for example, is this also comparable with, the kind of scenario that we are seeing in terms of the girth of that particular graph or the peak? Is this also comparable with the MEDIP-seq or the CLIP-seq based approaches, where we perform the cross-linking immunoprecipitation? And is it really the same? Because we largely work on the CLIP-seq, Rohit.
ClipSeq, okay.
Yeah, it's a cross-linking immunopresentation. The idea here is,
Yeah.
since our lab largely works on the long non-coding RNAs, so what we deem to look is to check whether or not any long non-coding RNAs, are known to be interacting with any proteins of our interest.
Yeah.
And then we ask a question: is it at a greater proportion, or do we get to see that long non-coding RNA largely upregulated in the interest? Okay, so that was
Yeah.
so. If you really, let's say, Shoma, I have a ClipSeq data, something of this sort. Can I just peep into this Washington University browser and see the similar peaks, and the density would be the same, isn't it?
Yeah, exactly. Like I have looked at RipSeq. I haven't, sorry, not RipSeq, ClipSeq. I have worked with ClipSeq, and I wasn't looking at non-coding RNA, but I'm pretty sure, like, if you know exactly where non-coding RNA is in the genome, the location of it, you can load the data and see it, and you can see exactly within your long-coding RNA if there's a preference for that protein to bind towards a particular region, particular motif that RNA has that would you can actually determine. So that's you can do through this. You can look at through the genome browser as well.
Wonderful. Thank you so much, Rohit. Yeah, please. Thank you so much.
Okay.
So,
All right. So
there is a small question, like I've kept it in the chat. Can you please try to answer?
Let me go.
If you want, I can ask that question. No problem. Sir,
Sure,
actually,
you can you ask the question?
So actually what happens during the evolution, whatever genes, there will be a duplication and divergency. Like whatever new genes we have in the system, one of the reasons is duplication of the existing genes and divergency. Now, even when we talk about transcription factors, a similar fate is happening. Like most of the transcription factors are evolved from pre-existing transcription factors.
True. Yeah.
There's a combination, some exon combination, something like that. They gave rise to a new kind of transcription factor. Now, when we are trying to use our antibody to target a particular transcription factor, what is the probability that we are targeting a similar transcription factor? Because there is a probability that we could able to antibody will recognize the say other transcription factor. That's all.
That's a great question, and that's actually a real problem in ChIP-seq. So I'll give you an example. There is a family of transcription factor called as IRFs. There is 9 IRFs. IRF 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay. If you're interested in only looking at IRF 1, for instance, but the antibody might, because of the reasons you've mentioned, they're so similar in terms of, they're basically kind of like homologs of each other in this case. So basically, that's actually a problem when you're looking at an antibody that might actually recognize IRF2, IRF3, and you're not sure. I think one way to actually get around it is it's mostly in like an in vitro setting, is you actually do a knockout as a control. Okay, so now you are looking at IRF1 ChIP-seq, and the control is IRF1 knockout cell lines. So all the peaks that you see in your IRF1 knockout are actually coming from either antibody that, like if there's a peak that is present in IRF1, but not in the IRF1 ChIP-seq, but not in the IRF1 knockout, are actual peaks. But if the peak is actually present both in IRF1 knockout and also in IRF1 ChIP-seq, you can probably say that is coming from that antibody recognizing some other IRFs, right? So that's how you kind of can come around it. But that's actually a real problem if you cannot generate those knockouts. It's very hard to raise an antibody that is very specific, because you have to basically make sure that that ChIP-seq antibody is really good enough that it's only recognizing a particular, IRF and not the others. Yeah, that's a great question.
Thank you, sir.
Any other questions?
Sir, I have questions. Sir, I'm from plant science background. So whether this server is useful for plant transcriptome data also, or it is limited to human or some mammalian system only?
So you can do plant biology with this chip. You can apply plant biology ChIP-seq. You can apply ChIP-seq to plant biology as well. The problem would actually be, first of all, finding a proper antibody, a plant-specific antibody, right, for your transcription factor of interest. And I think once you have that, I think the process downstreams is actually very similar. It doesn't matter what genome it is. If you have a very well annotated system where you know exactly what genes there are, a majority of these tools like motif analysis and gene ontology analysis, and these are kind of very developed morely for mammalian systems. So you might have some struggles there because you're trying to find out what genes there are that this transcription factor is binding, but you can't because those genes are not annotated. We don't know what those do in plant systems. We have never seen those motifs, so it will be very interesting, I would definitely say. But there will be a lot of challenges for the downstream part of the analysis, if because it's not very well tuned for the data sets that have been developed for the downstream part are not very well. It doesn't have a lot of plant annotations in them.
Okay, sir. Yeah, thank you.
Yeah. All right. So if there are no more questions, then I'll move on to this part. So now I'm going to tell you an example of how we apply ChIP-seq to interrogate a biological process. So this is actually one of the work that I have done here. And we were looking at something called as a microglia. A microglia is an immune cell. It's like a macrophage in the brain. And they actually maintain a homeostatic setting. That's where they're, growing, proliferation. That's like they're the normal state. What we discovered that there is a protein called COP1, and for the plant people out there, you probably know COP1 because it was actually first discovered in plants. It is actually involved in the photosynthesis process in COP1. It's really impressive. That's actually conserved all the way to mammals and serves a very important function. So COP1 is actually a ubiquitin ligase, which means that it actually puts ubiquitin chains on different proteins. And once the protein has those ubiquitin chains, they get degraded. That's a signal for their degradation. So that's how COP1 maintains a level of certain transcription factors. And in case of microglia, the transcription factor is CEBP beta. So COP1 regulates the level of CEBP beta in a homeostatic setting for these microglia. However, when you knock out COP1, what you see is that it can no longer, mark this transcription factor for degradation, which leads to accumulation of this transcription factor, which is because it's no longer getting degraded. Because of this accumulation of this transcription factor, this microglia becomes activated. It changes its morphology. It is secreting a lot of cytokines, and it is, in turn, really damaging the neurons in the brain. It attacks these neurons, and these neurons get damaged, and there's a possible link of this to neurodegeneration, which is a leading cause of Alzheimer's. And so we wanted to understand basically how the CEBP beta is binding, where it is binding in the genome. This was done in mouse brains. So where in mouse microglia or mouse macrophages this transcription factor is binding, how it is regulating, and what happens when you knock out COP1 and you have an accumulation of this transcription factor, how this actually leads to an activation of microglia. So to do that, we actually designed the experiment. I'm showing you this example for the bone marrow derived macrophages in mouse, but we have done this for microglia as well. So basically, we had two samples. One is a COP1 wild type setting where you have some amount of CEBP beta, and then you have COP1 knockout macrophages where you have a lot of CEBP beta. For this both systems, we are using an antibody against CEBP beta, so look for basically all the CEBP beta binding sites. Then we are also looking at something called as a histone mark, and you probably might have seen it. So histone marks, as I told you, these are the histones that are the histones which are part of the nucleosome where the DNA is actually wrapped. And these histones can get, they have these chains that come out of these, which can get methylated, acetylated, which actually determines, whether this DNA is going to actually move and open this region or whether it's going to close this region. So there's a lot of these modifications that happen onto these histones. One of them is actually the trimethylation of this H3K4, which means that the region is active promoter. Another is acetylation of this H3K27 chain of the histone, which means that this is mostly present in active enhancers. So this way, once you do antibody against these two, you can now figure out which regions of the genome are active promoters, which regions of the genome are active enhancers. So using these three, we did a ChIP-seq experiment, and here are the results. So I showed you this kind of a plot before. This is a CEBP beta peaks centered right here. We are looking at a 2 kb window around the peak. And I'm looking at the reads. I've aligned these reads to the peak. And each row here actually represents a single peaks. We found about 25,000 CEBP peaks around these two data sets. And as you can see, in the copper wild type setting, where you only have a little bit of CEBP beta, you can see there's so much more binding of CEBP beta that is happening in a copper knockout setting where you have accumulation of CEBP beta. And if you look at these particular regions and look at the promoter mark and enhancer mark, you can clearly see that there is not much of the promoter, this STK4 trimethylation mark here, but a lot more of this enhancer mark, which is actually increasing. And it's actually quantified here, where you can see there's so much more binding that is happening in copper knockout setting for CEBP beta, and not so much change in the promoter mark, but the enhancer mark in the knockout setting is a lot more, suggesting that these peaks that we are seeing is actually mostly coming from enhancers. So I took these peaks and I put it in that great tool that I was telling you about, and exactly it's what it told me. You can see that majority of the peaks that it's suggesting was actually 5 to 500 kb away from the TSS, coming up, suggesting that these are actually enhancer peaks. And if you look at the ontology analysis, the ontology terms that actually come up is response to bacteria, response to LPS, adaptive immune response, suggesting that these genes are actually regulating this CEBP beta is actually regulating the genes that is involved in some sort of immune activation, which makes which kind of explains why these microglia were activated when we deleted COP1 and there was an accumulation of CEBP beta. And now, if you look at the motifs, and I was telling you how each transcription factor has a preference for a DNA element that is very specific. Like in this case, it's ATT GCG CAAC, which kind of describes that this is a CEBP beta binding motif. And so you can see that not only it gave me the top motif that was enriched in the peak, but also gave me the different cofactors that CEBP beta binds to. And this is I'm plotting here is just the minus log 10 of the enrichment p-value. It kind of gives me basically what are the different motifs that are actually enriched. So this is an example of applying ChIP-seq data, ChIP-seq peaks, and basically once you have the peaks from here, I went on to look at what those peaks are, whether they're enhancer or promoter peaks, what is the function, and the motif. Kind of really gives you insight about what the biology might be going on for this particular transcription factor. Now, I think I only have 6 minutes left, so maybe I'll just quickly jump into the part. So ChIP-seq technology is limited by resolution. What do I mean by that? So imagine you are sitting in space and you're looking and trying to locate some signal on Earth, right? What ChIP, which is basically chip using a microarray, you will have a probably bigger region. You can detect a signal. You can detect like a sign that you're looking for on Earth, but you will have a very narrow, very, very big window of where that might be. You'll probably say, I see it somewhere between this big region. What ChIP-seq did is it basically narrowed it down for you and said, okay, I think the resolution is very clear. I think I can narrow it down to basically maybe this particular region over here. I see something, but I can't exactly figure out. It's somewhere between this big circle. There is another new technology, which is called as ChIP-exo, which gives you a near base pair resolution to literally figure out where your transcription factor is binding onto the genome. What do I mean by that? So when you get a ChIP-seq peak, it's usually 200 to 300 base pair. Now, 200 to 300 base pair is a very big region because you know that the motif that the transcription factor is binding to is probably 8 to 10 base pair long. So within that 300 base pair window, you're looking for something that is 10 base pair long, kind of like that. With ChIP-exo, you exactly know which 8 to 10 base pair region the transcription factor is binding to. So basically, that's the improved resolution. And how did they actually do that? So this is a protocol for ChIP-seq. Essentially, for ChIP-exo, the trick was actually using this thing called exonuclease, a 5' to 3' exonuclease. And what does the exonuclease do is it actually goes in the 5' to 3' direction, and it chews off the strand, one strand from 5' to 3', all the way until it hits the point of cross-linking. And it does the same thing in the reverse direction as well. So now when you get the reads out of it, the 5' end of this read in the positive strand and in the negative strand actually gives you exactly where the cross-linking has happened. And you can see, compared to ChIP-seq, this is a very narrow region where you can now really figure out where the transcription factor is binding. And to give you a solid example of this is transcription factor REB1, where I plotted both ChIP-chip, ChIP-seq, and ChIP-exo. And you can see, compared to ChIP-seq, which is shown in green here, ChIP-exo really tells you exactly within like the 10 to 20 base pair where the binding enrichment is. This is the enrichment that I'm plotting, the signal value for ChIP-exo compared to ChIP-seq and ChIP-chip. And so this is really has improved the mapping accuracy and sensitivity compared to the existing technologies. Okay. Now I want to move on to AttackSeek, but Prash, I wanted to get some feedback. It's almost 7. Should I continue on with the presentation, or do you think I should just stop and do the workshop?
Yeah, you can continue for the next 10-15 minutes, Rohit. We still have time. Yeah.
Okay.
7:45 is a good time for. I mean, another 23 minutes is a good time. I mean, 18 minutes is a good time, for hands-on. Yeah.
Okay. Okay. Sounds good.
Thank you. Thank you so much.
Okay. Yeah. Okay. So now I'll quickly tell you about what AttackSeek does. AttackSeek is not designed for any specific transcription pattern. AttackSeek is an orthogonal assay which tells you where the open chromatin is in your DNA. Oh, and open chromatin is basically, if you remember the first slide, when you had the +1 nucleosome and the minus nucleosome, and you had to have this open region, that's basically what open chromatin is. And you want to know which, and as if you remember, I told you for a gene to transcribe, you have to have that open chromatin region. This is kind of a proxy to basically say that that gene is active, that gene is transcribing because that gene is actually open. And so how do they do that? AttackSeek is actually a much simpler protocol compared to ChIP-seq, because in ChIP-seq, you can imagine you have to cross-link, then you have to sonicate, then you immunoprecipitate, and reverse cross-link. It just goes on and on and on and on, and takes a very long time. AttackSeek protocol is very simple, very fast. It's basically, as I mentioned, these are the nucleosomes here. This is a DNA that is wrapped around it. This is a region you're interested in. They use a TN5 transposome, which basically nicks or cuts these open chromatin regions, and as well as cutting, it actually also puts in these adapters. So now you can basically isolate these DNA fragments that already have these adapters, amplify and sequence it. That's it. So that gives you a really, gives this, since the protocol is so fast that people who don't need a lot of experience optimizing the protocol and actually getting through it can actually get it done very fast. And that's why this actually, got picked up. This protocol AttackSeek became so popular because it was so easy to do. Another advantage of AttackSeek is it can be done with very less number of cells. So for ChIP-seq, you at least need 2 million cells, right? And sometimes, if you're looking at patient samples, for instance, you will not get 2 million cells. That is impossible thing to get. You can grow your cells and you can, harvest cells, and you have some in vitro system, you can get those cells and you can do it. But if you're looking at in vivo system, AttackSeek is very powerful because you can actually go down to even 500 cells and perform AttackSeek and really get good signal. So here I'm showing you one of those genome screenshots where I'm comparing the signal strands for AttackSeek using 50,000 cells. This is DNA's hypersensitive sites, which is basically another enzyme that cuts in the open chromatin region. So you do get basically the same signal where the region is open, where the chromatin is open, but that's done using 1 to 50 million cells. FAIR-seq is also a very similar protocol. It's formaldehyde enriched regions which are open chromatin. Again, 1 to 50 million cells. And the lower panel is actually showing ataxia using 500 cells. And you can see with 500 cells, it's pretty much giving you all these same signal strands that you can actually get from a 50 million cells or even 50,000 cells. And that's quite impressive. And that's why ataxia became so popular, because you can interrogate, these open chromatin in the region and with a fast protocol and with very few cells. That was the really powerful approach, and kind of threw other technologies outside the bucket. So the analysis workflow for Ataxic is pretty much very similar, except in Ataxic, you do not have any input control because there is really nothing to compare, because you're not really enriching for a specific transcription factor. And again, with Ataxic, you should do this experiment replicate. And so the analysis workflow is kind of very similar. Like you have these raw reads, you do the alignment, use the peak calling. You can use Max2 or GPS for this. Basically, the same peak calling tools are available. This is a case control analysis. So, for instance, if you do an Ataxic in an untreated versus a treated setting, you can kind of do those kind of analysis. You can look at where your Ataxic peaks are, whether they are enriched in promoters or enhancers, or in a intergenic region, in exonic regions, intronic regions, things like that. You do the motif analysis, because at the end of the day, you can imagine for a treatment versus control setting, let's say you're looking at a homeostatic microglia versus an active microglia. You want to look at what changed in terms of open chromatin region between these two settings. You can figure out those differential peaks and then do a motif analysis to figure out what motif was underneath those peaks that actually changed that actually changed the peaks. So that's kind of the analysis that you can do. I think the real power is actually what people are figuring out is associating SNPs, because so far with all the GWAS analysis that we have done, that has been only limited to the exonic regions, right? Because if you have a mutation in your gene, you can then basically predict whether it's going to alter the protein or not, and then say, okay, looks like it's going to have a dampening effect on the protein association with the disease. But we could never evaluate the SNPs that fell in the non-coding regions because we don't know what they do. Now, if you see if there is a ataxic peak and underneath that ataxic peak, if that ataxic peak is in a non-coding region, and you see there is a SNP that is associated, and you can associate that ataxic peak to the nearest gene or to whatever gene, then you can actually now have some functional link to these SNPs that you could never associate before. So that's actually the powerful approach that's been currently used right now to kind of associate these non-coding peaks to some sort of a biology. This plot, I wanted to highlight this because this is one of the QC metric that people use. So if you look at the fragment length distribution for an ataxic, you can imagine that when you use this transposome, it can nick over here. So you can get the length of the fragment that would sequence would actually be about 147 base pair plus minus 20 base pairs, because you're getting one nucleosome. You can also get nicks where you can get two nucleosomes or three nucleosomes. So there you can see that when you look at the distribution of fragment length, it should be cyclic, where you have enrichment for one nucleosome, two nucleosome, three nucleosome. So kind of like this, where you have a stronger enrichment, like stronger peak for, let's say, a transcription factor, but then you get a one nucleosome, dinucleosome, trinucleosome kind of a pattern. This gives you sort of an indication that your ataxic protocol actually worked, and you have this sort of a fragment length distribution that kind of mimics what you should be expecting. So kind of really going where ataxic workflow is very similar to the ChIP-seq protocol. So there's not really much I can tell you here. But I wanted to give you an example from how people have applied ataxic to learn about biological processes. And I wanted to highlight this particular paper here. This is a published work, but it was at least an eye-opening paper for me, because it kind of tells you that each, we all know that each cell has its own, chromatin organization. And when a cell differentiates from a stem-like to more of a differentiated cell type, that chromatin organization really changes because some genes go up, some genes go down, and those are the genes that define how the transcript, how that stem cell would actually differentiate or to take one trajectory versus the other trajectory. And they apply this to hematopoietic stem cell differentiation, where the hematopoietic stem cell, which can self-proliferate, they go into these progenitor cells, and then, they figure out they either go to a lymphoid lineage or a myeloid lineage, and then they either end up becoming CD4 T cells, CD8 T cells, B cells, or NK cells in the lymphoid, or monocytes, granulocytes, thriocyte, and megakaryocytes in the myeloid lineage. And if you look at so this is basically the isolated, they fact sorted all of these different cell types, and then they did ATAC-seq on each of these cell types. And that's what they're showing here on the y-axis. On the x-axis, they're showing the genes that are actually important in playing a role in maintaining that particular lineage. So for instance, GATA2 is actually known for playing an important role in the stem cell proliferation and maintenance. And you can see for GATA2, it's not the chromatin, the CD4, CD8 cells, very specific. You see this peak that is very specific to only the stem cells. Similarly, CEBP beta seems to be only present in these monocytes and not any of these lineages. BCL11B seems to be very specific to CD4 and CD8 T cells. BLK is present in these stem cells and also in these NK cells and B cells. So what this tells you is that these chromatins or these ATAC-seq peaks are very cell type specific. And we can use this information to actually classify the cells into different types. And that's what they did in this paper. But what I wanted to highlight in this particular example was they're looking at this gene called TET2. And they are looking at, each row is showing an ATAC-seq signal for different cell types and corresponding RNA-seq signal. Now, if you just blind look at this data and you just look at the transcription start site, it looks like the transcription start site looks like the STET region is open in all of these cell types. However, why is there such difference in the RNA-seq for this gene? And that is basically because that this gene's expression is actually controlled by enhancer-specific regions. So, for instance, there is this NK cell-specific enhancer that controls the expression of TET, and that is only present in NK cells and a little bit in CD8 T cells. There is this stem cell-specific enhancer that is only present in stem cells that actually is controlling the expression of TET2 together with the gene, together with the promoter accessibility here. And then there's a lymphoid-specific enhancer. So, if you just try to figure out the if you just try to look at the RNA-seq expression just by looking at the transcription start site, you probably can't figure it out because the expression of this gene is actually not only controlled by the promoter, but also cell type specific enhancers. And I think that's the critical part here. Because imagine if you have a mutation, right? Let's say the mutation is present here. You can just say, okay, this mutation is there, but I don't really know how that really impacts. But looking at enhancer, you can say, okay, I think it only impacts NK cells because what will happen is if that mutation disturbs this open chromatin, it won't really affect the stem cells. Nothing would happen here. But in NK cells, that would actually impact the expression of TET2 genes, and it will be very NK cell specific. So that's really where the biology comes in, when you're associating these enhancers, these genes together with the mutations. So in interest of time, I think I just want to highlight that ATAC-seq technology is actually really moving at a very advanced, really fast space. And not only you have, this was all bulk level ATAC-seq data, but now you're doing ATAC-seq at a single cell level. And the first paper that came out in 2015, they did single cell ATAC-seq with 254 cell, basically getting the ATAC-seq signal strengths from each row here represents each single cell. And this represents sort of just a composite of all of these 254 cells. And this was the first study that showed that you could do an ATAC-seq analysis at a single cell level. But more than that, now you can combine the ATAC-seq with RNA-seq at a single cell level. And so I'm not sure how people are familiar here with the single cell level analysis here, but each dot in this particular t-SNE map shows a single cell. And so not only are getting the RNA-seq expression, the gene expression information from one single cell, but also the chromatin accessibility from that particular cell. And as I was mentioning before, now you can do a much deeper analysis where now you can look at chromatin accessibility in each cell type. So you can say this region is only open in monocyte, and it seems that there is a SNP that is associated with this. This would really justify why this disease, is so and so, because this is a monocyte specific effect of the SNP, and why it is not in any of these cell types. And lastly, it's not just the single cell technology. All of these technologies that I mentioned before were all bulk level. So you're doing it, you take the whole tissue, you grind it up, and you do it. You look at the population level effect. All of these technologies are now looking at individual cells, and ChIP-seq is also moving in that direction. And there has been some papers where people are doing single cell level ChIP-seq. I have never done this kind of analysis, but this, all of these bulk level technologies like RNA-seq, ChIP-seq, ATAC-seq are now moving towards more of a single cell level assays. So that's all. I have put a link to all of these resources, all the different websites that I've mentioned, the tools, the packages, and whatnot that's in there. Feel free to browse it. You have this presentation and the slides I shared. And I just wanted to thank my advisor from Penn State and my current colleagues at Genentech for the feedback. And I'll take any questions if you have, and maybe we can move on to the workshop part of the analysis.
Sir, I have one question.
Yeah.
So, any other method to validate the results, whatever we are getting from this ATAC-seq and other seqs? Like, do we have only this method, or we have alternative methods to validate this particular results from this particular experiments?
So, I mean, what do you mean by validation? Can you talk about that a little?
No, actually, whatever results we are getting, like most of the time, this is all very large set of data.
Yeah.
So now sometimes we need to validate this particular result. Let us say when we do NGS for the human genome, we will get lots of data, and we try to validate that particular information with the help of Sanger sequencing.
I see.
So similarly, do we have some other kind of technique by using which we can validate the whatever results we are getting in ATAC-seq or other ChIP-seq things?
So you can validate it, but you can use some sort of like orthogonal approaches to kind of looking at the same genes. So for instance, if you're only interested in one gene, then you can do, if you found out that there was a motif that was, if you found out a peak that was there for that particular gene, you can do ChIP qPCR and basically see whether that is enriched or not. You can make mutations in that particular region to see whether that disrupts the binding. That gives you more confidence that, okay, that is the motif where my transcription factor of interest was binding. I made a mutation, and now I lost that binding. You can do qPCR on the just particular. So you can do the validation for a specific gene of interest and look at whether the discoveries that you made here and modifying them. You can use CRISPR and knock out that whole motif and see how now the binding is disrupted, how that impacts the open chromatin, how does it impact the expression of that particular gene. So you can do those kinds of things.
Thank you, sir.
Okay. If you have more questions, feel free to let me know. Otherwise, I'll move on to the workshop part in the 45 minutes or so I have. So basically,
I have a question.
Oh, yeah, sure.
Just a minute. Yeah. So, would you say that this EdTech-seq is better than the previous one, ChipSeq for plants? The same question that Vipul had.
Yeah.
Is it a better
Uh,
option?
I mean, I think limitations really come from, I don't think the technology. I think you can apply ChipSeq, you can apply TaxSeq. I think the limitations really come from the inability to understand the biology, the analysis part, because the tools will work fine. I think that the tools are agnostic to what genome you give them and what this. But I think the downstream tool is where, like the motif, for instance, let's say you did this and you can run meme because meme will give you a motif. Now you've got the motif, but the motif database probably will not have a good collection of plant motifs that are most commonly, accessible, that are most commonly found. So you will get a motif, but then you won't know exactly what this is. You won't know what it does. If you do an ontology analysis, maybe, and I don't know a lot of plant genetics, plant genomics. I don't really know if there's a really good annotation for ontology purposes, like if you have a set of genes, then will it give you function or not. So, like for instance, great is only available for human mouse. So you can't even do zebrafish or any other model organism. So that's where the challenges really come in. You can do epigenome browser and you can visualize the tools, because I think this is open to all kinds of genomes. So that's where I think the limitations would really come in terms of more downstream analysis, where you're really trying to understand the function, things like that. That's not because of the technology. That's mostly because of the annotation that is available.
Right. Thank you. Thank you, Rohit.
Yeah. So for the workshop, I'm going to be focusing on these three things. And before I go there, I wanted to just, just make sure everybody's on the same page. And if you're in bioinformatics, you probably have come across at least one of these formats. I feel like since the last 20 years, there's like one file format that has come up, every year. That's how, everybody who designs a tool comes up with their own set of formats. And so it's hard to keep track of. ENCODE has a list of commonly available, formats, and you probably must, aware of like FASTQ, FASTA formats, VCF, GFF. I don't know if you guys know about this bed format, but this is the most common use for ChipSeq. And this is basically a format that has 6 columns. So you guys can still see my screen, right?
Yes, sir, we can see that.
Okay. So I have put in a bed file here. This is a C beta, ChipSeq peaks that I mentioned in the earlier slides, and this is basically how a peak file looks like. So you have chromosome.
In bed file where you have opened?
Sorry.
In which one you have opened bed file?
Oh, so this is that workshop material that I sent in the link. So let me see. So in the chat, I shared a link. This link. So if you go to this Google Drive link, there is a lot of files here that I have put in. You can download all of this, and that's where the bed file is.
I'm not asking about the file. I'm asking about in which tool you have opened the bed file.
Oh, this is a terminal. Yeah, it's just terminal. I haven't opened any tool yet.
Can you please share me the drive?
How to go to terminal?
No. The link you have shared already.
Okay.
Because I got disconnected for a minute, so I lost previous.
Here. I'll send it again.
Thank you so much. I got it.
Yeah. Okay. Can you also see my screen? Yes.
Yes.
Okay. So this is the bed file format, which is basically has these 6 columns: chromosome, start, and the name of the peak and some sort of strength of that particular peak. So you can see that this is all the places where CEBP beta binds in a COP1 knockout setting. That's what I named. And this is what you will get an output of max2. So that's where I'm starting from. So the parts that are upstream of this pipeline, like mapping and peak calling, can be done in Galaxy. However, I can't really show you that to you right now, but I can. That's why, because I just wanted to start with more of a downstream analysis that was easy to show. And so once you have these peaks, that's how kind of basically a peak looks like. And for this particular study, I had about 25,000 peaks. No, this is just 1,000 peaks just for the analysis purposes that I put here. Okay, so that's what I wanted to show you about the different peaks or different file formats. Another thing I wanted to tell you about is something called bed tools. So bed tools is a really good set of tools for doing genomic arithmetics, right? You have files like bed or GFF or VCF, and sometimes what you want to do, you probably might have heard about this tool, but sometimes what you want to do is you want to look at regions that are intersecting, regions that are separate, like all kind of these, like you want to merge the files, you want to separate the files. This is a really good tool to perform those kind of arithmetic operations. And so, for instance, if you want to remove, let's say, the blacklisted regions from your peaks. So this is your peak. And I've also put in here, let me just zoom in so you can actually see this much better. Okay. So I've also put in here the blacklisted regions. Did I? Maybe I did not put the blacklist regions. Let's see.
Excuse me, sir.
Yeah.
Hello, sir.
Yeah.
Yes, I have one question. Can we use these things in Galaxy also, or we have to use it only in the this terminal which you are using right now?
Bed tools you can use in Galaxy. They should have Galaxy tools there, yeah, because I think that's a very common set of tools. So you can actually use bed tools, but you can also use. I would rather do it on my terminal on my server because it's much faster and it's just for simple operations like I want to subset my peaks. I want to do an intersection between the peaks, things like that. I want to. So I have put in here. Let's see where it is. The different bed tools commands. So you can actually, try to do this when you have time. So this is basically an intersect bed command. So I want to remove the blacklisted region. So you can download the list of blacklisted region for mouse and human. They have separate list of regions. You can download it from here. And I want to remove these regions from my peaks. Okay. For that, you would use something called intersect bed. And I think I have installed on my computer. And if you do this, you would get like a lot of, this is intersect bed, what it does, and how you should be using it. And for removing blacklist regions, I actually have this here. So basically, the command that you would run is say intersect bed -v means give me all the regions that are present in my file A that are not part of file B. So you will say, give me only the regions that are not here. So I would say -v, -a, my file one bed file, which is my CE beta chip seq file, -b, which is my blacklisted file. And you run this, and then you would save it to a different file, which would be basically all the regions where I have taken out the blacklisted regions. Similarly, you can you can convert this bed. So, the bed file here is essentially a list of coordinates, and you can extract the sequence underneath those peaks using a command called faster from bed. And for the faster from bed, you would say faster from bed, my input, and this is your reference genome. So I did not share this file because it's a really big file, but you can download it from Ensemble. And this is basically the sequence of the whole mouse chromosomes. And so you say, this is my reference. This is my bed file, which I want to download the coordinates for, and save it as a FASTA file, which is basically the file I want to use for my downstream analysis. So once you do that, you would get a FASTA file that looks like this. which is basically these are the peaks that I was showing you before in the bed file, and it extracts all the DNA sequence that was underneath this particular peak. Okay. So now you take these peaks and you go to a tool called Mean. So it's Mean Suite. This is the online tool I was telling you about. And the motif discovery tool is either Mean or Mean Chip. Mean Chip is actually used for when you have a really big data set. You have 25,000 peaks, and each peak is 100 base pair long. That's when you use Mean Chip. You go to Mean, and then you basically, you can choose the different parameters. I'm running it in a classic mode. And it says upload your FASTA file here. So you upload sequence, you choose a file. And in this case, I have the FASTA file here, which I will choose. It will upload it. You can tell it how many motifs do you want. You can say 3, more than 3. You can just put it here. I would put in my email address here because this tool actually takes a lot of time to run. And so you cannot just have it, run in the background. I mean, you can have it run in the background, but it takes a lot of time for it to run. So once you submit the job, you put in your email address and you just say start search, and then this will submit the job, but it will take a lot of time for it to run. So I knew this was going to take a lot of time. So I actually had run a job previously. So I'm just going to show you what happens when you do that. So this was the CEBP beta peaks. And so that the job runs fast, I just use the top 1,000 peaks to do this analysis. And what you get at the end is just basically this HTML output. So you can click on this output and you get something like this. So you can see you get a bunch of motifs that are enriched, and some of them kind of make sense. This is a short enough motif. This is something that is really long, and who knows what this even is. This is just a TTTT to repeat. So let's say this looks interesting to you, and you want to know what this motif is, this TTTGC motif is. So you go to this button here that says submit or download. So you can download this motif here, but you can also submit this tool to this another tool called as TomTom. And so you click submit. It takes you to the TomTom page. You can choose what database you want to run it against. I mean, this is a eukaryotic DNA, but you can also choose prokaryotic DNA. Then you can say, but this is vertebrate. Let's see if there's any plant thing here. This is basically choosing the database of motifs, and you basically say start my search. This runs for a little time, and I have already run this, so I'll just go to the results directly. And you can go to the TomTom results, and it basically says for the motif that you queried, oh, it looks like it's a CEBP beta motif. So this is a sort of a validation that okay, I use an antibody against the CEBP beta, and I'm getting the CEBP beta motif. That means the experiment is valid. If I was getting CTCF, then maybe my antibody is recognizing something else, or it is very non-specific. And on top of that, other than top motifs, you can also get, from your motif analysis, you can also get generally other cofactors that are actually enriched with CEBP beta. So these are the top 3 motifs, but if you repeat this analysis with, let's say, more than 3 or 6 or 7 motifs, you can also get some of the cofactors that are actually enriched along in your peaks. So any questions so far? I know I went a little fast on that, so let me know if you have any questions about basically going from your peaks to your FASTA files to doing a meme analysis. Okay.
So can you just show how you get the map reads, the first step?
So the map reads is basically, you would get it in using Galaxy or any other tools if you're familiar with Bowtie or BWA. You would take your FASTA-Q files and you would run it through those mapping algorithms. That's how you would get your map reads. And then you can run max2, which is also can be done in Galaxy, or you can do it on your server. You download this tool, you run it on the BAM files, and that's how you get the max2 output, which is these peaks that I've been showing you, which is basically looks like this, like an enrichment across your genome. Okay. Yeah, so the next thing I wanted to show, basically I'm trying to reproduce this result here, and I'm trying to see whether the CV beta peaks that I get, so this is the motif that I just showed you, the TTG, AAC motif. That's how we recovered the motif for CV beta. Now I'm going to show you how to do the enrichment analysis and look at what this CV beta motif, whether it's promoter motif, enhancer motif. So for that, you go to this tool. I have a link for that as well. You go to this tool. And as I mentioned, this was mostly done for human and mouse, so that might be difficult for plant genomics. But you choose your reference genome and you upload your bed file, which is your C beta peak file. And you basically click submit. That's all. And this takes a little bit time. It runs through because I have a lot of peaks here. But I think I put in the results here because I knew this is going, it already ran. Great. So you can see it gives you, first plot it gives you is basically the gene region association. So it tells you, okay, the top 1,000 peaks that I put in, majority of seems to be enhancer peaks. You can see that these are 50 to 500 kb away from the TSS. So kind of, sort of suggesting that these CEB beta peaks that I've gotten are majority of them are enhancer peaks compared to the promoter peaks. And then you go down, you get this, the gene ontology analysis, which ranks all of these top terms that you actually get. And you can go to this column that says visualize this table. And you can visualize this as a bar chart. So this is the kind of the plot that I was showing you before. It plots the p-values on the x-axis, and it plots the terms on the y-axis. And you get something like this, where it says, response to bacterium, response to adapt immune systems, kind of suggesting that these peaks are regulating genes that are involved in immune activation. So this is kind of another way for you to kind of understand what that particular transcription factor of interest is really doing in your system. And here it's plotting these p-values on the y-axis. So the stronger the p-value, the more association of that term is to your genes of interest. So this is really easy to use tool. All you need is your set of peak files. You upload the peak files, you choose your genome, and you get really these analysis that you can really understand what your system is doing. And the last tool that I wanted to give you a demo of was actually the WashU Epigenome browser. This is the link for the WashU Epigenome browser. And so you can go here and it first ask you to choose your genome. So for me, it's a mouse genome. So I'll go and choose an MM10 version of the mouse. And you can see it already, gives you basically where the genes are and some of the other annotations that are already present. Now, this is actually a little bit challenging part to actually go from your BAM files to the files that you can actually upload here and look at it. And so to assist you with that process, I have left some notes here. So notes are in this file, bedtoolcommands.txt. And this basically tells you how you can go from your BAM files to something called as a bed graph file, which you can then visualize on your Epigenome browser. So again, you would need bed tools for this purposes, and you would need something called as HitSlim tools, which is part of the SAM tools package. These are very commonly available tools that are easy to download and install. And so first step is actually using bed tools. You convert your BAM files into a bed graph file. which is, BAM file is a binary file, and bed graph is basically a chromosome and some sort of chromosome position and some sort of an enrichment value that you get in that column. And then you take the bed graph file and you sort of compress it using this htslib program. So you zip it and then you index that using these two commands. So I'm not going to show you this online because that takes a bit of time, but I wanted to have these commands with you so you can practice it. I have a file for you here. So this is the BAM file here. This is a very small BAM file, so you can actually practice it. And once you make this BAM file, you would get this bed graph file, which then you can compress. You'll get this GZ file, and then you can index it, and you can get this tb.tbi file. So these are all the files that are already in there for you to sort of practice. And once you've downloaded and installed those tools, you will be able to kind of work it out at your own pace. So once you have this bed graph files and this indexed file, now you can actually upload those on this Epigenome browser. So the way to do it is you actually go to this tracks button over here, and you go to the option that says local tracks. And within the local tracks, you choose the tag type, which in this case is bed graph, because that's the file that we have created. And so this bed graph, you not only need a GZ file, but the indexed file. So you have to choose your files. So I'm going to choose my wild type bed graph file, which is a GZ file, and the indexed file. So these two together, you press shift and you press them together. So this is a wild type file. Then I'll go here again, and I have a knockout of those knockout data set as well. So this is a copper knockout, the bedgraph.gz and the bedgraph.tbi. I'm going to choose that as well. And so this is because BAM files are really, really big. So I've only subsetted the BAM file for a specific region of the genome. So I'm going to go here and I'm going to put this coordinate here to show you how this actually looks like. So this is a CXCL2 region that I wanted to show you, because in the knockout setting, we saw that there was an enrichment of binding of CEV beta to this particular gene of interest. And this is a cytokine, a chemokine actually, and so kind of associates with an activation state of the microglia. So what you would see is this blue tracks suggesting that this is where the CEV beta is actually binding. There is some binding at the promoter, but looks like a majority of this binding is happening at the enhancer. However, at this point, it just looks like it's basically the same. It's because we haven't really adjusted for the y-axis here. So you can see that the max here is 142 and the max here is 23. So if I go here and I adjust the y-axis and change it to fixed, and I make it, let's say, 142. So I'm basically looking at this track on the same y-axis. Now you can see that the CEV beta binding at this particular enhancer site actually increases quite a lot in the copper knockout setting compared to the wild type setting. And similarly, even at the promoter region, you can see that's the case as well. This actually correlates with increased expression of CXCL2, which is one of the hallmarks of an activated microglia. You can use this button to kind of slide, things. You can zoom on using these buttons like +1, +5. I mean, I don't know if you have ever had experience with any of this genome browser, but you can zoom in, you can zoom out using this -1 button. So you can, slowly zoom at the +1. You can keep on clicking until you zoom out, to the max. And now you can actually clearly see that this is a CXCL2 gene. This is a promoter region. There is such a strong enrichment of CEBP beta binding here compared to the wild type, suggesting, like not only at the promoter, but at the enhancer region, this gene is regulated by CEBP beta. And when you don't have carbon in this setting, the accumulation leads to binding at these sites. So that's pretty much, I think, what I wanted to cover. So far what I've showed you is how to basically regenerate this figure from the paper, where you can look at the enrichment of the transcription factor C repair binding and look at all the different cofactors using the FASTA file, getting from the starting from the bed file, getting the FASTA file, and doing the motif analysis, using that bed file to look at the ontology analysis and the distribution of those peaks, and concluding whether those peaks are promoter peaks or enhancer peaks, and looking at the ontology of those peaks. So there's a lot of data set that I've shared with you, and also together with the data set, I've also shared the analysis. So what kind of output you should expect. So, this is the bed file for both the wild type and the knockout. I have here how the great output should look like. So once you're following along in your own time, you can see whether you're actually getting the same outputs or not. I've shared the FASTA file. This is the blacklisted region that I've shared with you as well. This is the meme results, the indexed files, and also the PowerPoint that I was just going along with here today. So feel free to practice at your own pace, like trying to just figure out how to do this downstream analysis, and feel free to reach out if you have any questions. Yeah, that's all, and I'll stop sharing. If you have any questions.
Thank you, sir. It's a very good lecture.
Yeah, thank you.
Sir, can you show the deep tool analysis also?
I can't show the deep tool analysis because that would require like writing a script using those functions and running the script. So I would recommend deep tool has a really nice, like I'll show you, like it's a very easy to use. It's basically like you plot heat map. It's kind of like running bed tools, and it gives you like all of these commands that you can actually use. And like there's an example usage where you can go and create these files. How do you go about starting from the BEM files to creating these plots? It's a really, like there's a lot of commands that you can just really follow along and create different kinds of plots. It requires some level of understanding programming, but wait, I wasn't really showing you my screen. Sorry. Let me just share quickly. Sorry about that. No, not this one. Stop sharing. Okay. So this is the Deep Tools package. Can you all see my screen? I think you can see it now. It has like a step-by-step way of how do you install the tool. And then this example usage is what I found really useful. You can actually go in here and you can see different commands that you need to use to kind of generate these plots. There's like a lot of plots of correlating different data sets and a lot of QC plots, the fragment size distribution that you would need to use, a lot of QC metrics, and how to create, at the end of the day, like all the different plots of enrichment. A really nice package I would recommend. And this is probably what to do on your server. You can't really do it on Galaxy, but that's like some of the parts of bioinformatics are kind of more programming intense, and that's I think I would say deep tools. So you would have to have some sort of understanding of programming. Any other questions?
Wonderful talk, Rohit. Thank you so much. You have taken us to a completely different world altogether.
Thank you.
I know very, very few people are working in these gray areas of attack-seek and these things, chip-seek. Not many labs are really privileged in India yet. For instance, accepting some labs like IGIB or maybe NIBMG and CCNB. LCBS, they are doing, but the data that they are probably obtaining is not really again up to the mark. That's because we don't get the clinical data. As you know, the real challenge here is we don't get the clinical. We don't probably collaborate, with clinicians and hospitals directly. That is a kind of a great challenge. That is the only lacuna in India. And even I would say single cell RNA-seq is also at its infancy. It's certainly a kind of a hype, I would still say. People, unless they come to a bulk RNA-seq data, do it on at least on 50 or 60 companion of those particular phenotypes, and then jot down to a single cell thing reported. There's no this thing. It's very unfortunate thing, but over the years, I've seen over the last 3-4 years, there has been a big brain drain in India. I'm not sure if you're really following up because you've never been in India over the last few years. But several people are moving back to India, bringing out those wonderful technologies. I'm sure it will be a bit this thing.
No, I think it will definitely pick up, and I think I've seen a lot of papers coming out of India where people are now using a lot of these NGS technologies, which is really like I'm really excited for that, and it's really nice to see that this is coming, and that's why I wanted to kind of share some of these experiences with the people here in case that's where the direction they decide to take, that it should be like it will be great for me to kind of share these experiences and have an opportunity to talk about these different analyses that are out there. So thank you so much for giving me this opportunity.
No, not at all. And friends, I hope you'll appreciate the fact that Rohit has a Cell paper, a Nature paper, whatnot. He has carved out a wonderful niche over the years. When was the last time we met Rohit? It was only in 2009, is it? Or
I don't think we have ever met actually.
we
have always had. I have. I need to come to Kerala.
As I was telling you, this time we're organizing INBIX in VIT. INBIX has been a kind of a great conference events which we have been organizing over there.